Hi, in this video, we'll perform analysis regarding some of the basic experimental designs which are very much used in agriculture, forestry, and allied sciences. For that, we'll be using some of the libraries which are very uh, frequently used and highly recommended for analysis of variance, experimental designs, some factorial or incomplete block designs. So the first one will begin with the DOE bioresearch, AgroR, then agricolae. So if we will go to this console and we will write DOE bioresearch, so here we can find the descriptions of this library. It is a wonderful work done by this Raj Popat, who is a maintainer of this package. And this design DOE bioresearch is recommended for the analysis of experimental designs in biological or any kind of a research. We can perform some transformations like arc sine, log also, and some square root transformation, which are used whenever the data is not normally distributed when performing the experimental design. If you'll see, click on this. So we have, suppose, an example of factor of here. If we will copy paste it, we'll simply copy this and paste it like here. So you can see the original values are this, and these have been transformed to the arcsine transformations. Then if we'll go to the index also, then go to the log transformation, we can do retransformations also. Again, we have a vector here. We simply have to write log transform form, then write the name of the vector. So we have the log transform values, and similarly we can have some square root transformation values also for any vector. So for that you have to use these functions which are available in this library. So we have these transformations. These are usually used to stabilize the variability of that. So as far as the CRD is concerned, the most flexible design, for that we have a data set here which is available as an example. We'll copy it and we'll see what kind of data set it is, since the data is like this. If we click on this enter, so we have a data. Usually we used to feed the data on Excel. We can use this option, import option, so that we can import the data, or we can use a normal this read dot table for recalling or importing the data. But how you are going to use the data, and how you have to uh, feed the data in your Excel sheet, for that this is the demo, and this is the way you have to put your data in the Excel sheet. You will start with the first column should be treatment, then followed by the rest of the variables on which you want to execute this particular design. For that, we're using a function which is by default present. We have a name of a data, and we are interested to see to evaluate these 7 treatment with respect to this variable. Since the position of this variable is on second, we have write only 2, and if we want to both, then we will put this sign followed by this. Then we'll click on this enter. But before that, here this one, it is very important. It is used for multiple comparison analysis, which is the part of post hoc analysis. One, if you write 1, it will perform the least significant difference, and if you use 2, it will perform the Duncan's multiple range test. Then if we will click on the enter, we will have the results for this. We have ANOVA traditional, we have the values for normal T-Shapiro test, and we can find out here these are all non-significant. For the first variable, for second variable we have ANOVA, and we have a normal data. Since its p-value is greater than 0.05, it is non-significant, which is very highly recommended for testing the normality. Then we have the different group comparisons. Very quickly we'll move to the next design, that is RCBD. We have an example here also. We'll copy it and let us see the structure of this data, which is by default. This is the way how you have to put the data in this format like this. So first you will write the name of your variables on which you want to evaluate. Then you have to put the block variable. Here it is named as application, then followed by the treatments. Then how we are going to execute it. Say for example, we'll copy this and we'll use the default function followed by the name and the position of the variable in which you are interested. Then followed by this 2 or 1. In this case, if I write 1, it will perform least significant difference like in the previous thing. So this RCBD is a default code, default function rather. And in case of CRD, you have to simply write CRD followed by the name of the data, define the variable's position, then followed by the name of the data again. Then if you are having a factorial experiment, Then for that we have a factorial data which is by default present in this library, that is factorial data. So this is the factorial data, and if we want to view this, we will simply write this capital view. So we have a data and we have to feed the data in this like this format. First we put the name of the application, then the name of the first factor, second factor. Since we are doing two factor this factorial experiment in CRD for two factors, you have to put the data in this format. Then if you want to perform the analysis, for that you have to use this code where you can replace the name of data set, the position of your variable, and again if you want to do post hoc, DMRT, or multiple range tests, whatever the test. So for example, we'll write 2, we'll do the DMRT, we'll click on this enter. So we'll be having an ANOVA here for both these factors. For we can see whether they are significantly different from each other or not. We have means for the first factor, we have means for the second factor, and similarly we have a means for interactions also. So similarly we can perform the factorial experiment with three factors. For that we again using the same data set, and for that we will use this function. Okay, we'll click on this and press on the enter, and we have the results for all these. Now we have a 1, 2, and 3 factors here. Similarly you can do the analysis for factorial experiments also. So if you are interested to perform the split plot analysis also, you have a split data also, then you can perform the split plot analysis also, again using a different data set. Say for example the data is split data, and you might be having the this main plot or subplot where both these factors are assessed with unequal precision, main plot with less and subplot with more precision. You have to put first decide the blocking variable, then main plot, then subplot, then use variables which are supposed to be in your data set. For that you have to use this function. It's a very wonderful work done by the maintainers of this library. Simply put, use the default split plot code, then put the name of your data, position of the variable in which you are interested to evaluate, then write the name of your blocking variable, then your main plot and subplot, and the post hoc value you are performing LSD or DMRT, then click on the enter. So we have the analysis for this split plot as a split plot design. Here you can see actually this is the output of this. Sorry, this we have log, we have results for source of variation of main plot, and here you can see we have two errors, EA and EB, which are usually in the output of this analysis of variance. Similarly, if you are interested to perform some other kind of analysis which pertains to this factorial or single factorial, you can do another library which is highly recommended, that is known as agricolae, which is a very important library which is usually used for statistical procedures for agricultural research. And if we want to perform, say for example we want to do the basic LSD or DMRT test we want to do. Say for example if we want to have we have a data that is known as potato data. So let us see the structure of this data. We will write view, then potato. So we have these different viruses here or different treatments here, and you have a response variable, and you want to perform this kind of an analysis. For that you can use this function like this. AOV is the default function, yield versus the treatment, the name of the data, then lsd.test is the default code for performing the least significant difference, then the name of the function, then the treatments, then click on the enter, then we will write simply out. So we have the results for this also. So we can also use the DMRT also here, Duncan's multiple range test also, which is here, Duncan test. So again we're using the same thing, but here you can see we can make the plots also for this like this. We'll copy this. You can adjust or change the name of the data here as per the name of your data set. So we'll be having the plots also, we'll be having the DMRT post hoc analysis. Apart from this, what we can do in agricolae, we can perform this various types of breeding analysis. Say for example, we are interested to perform a line tester. For that we have a line tester data also. We can perform various kind of analysis, like we want to do L into T, line to tester. It makes the line into tester analysis. It also estimates the GCA and SCA, that is general and specific combining ability, and line tester contributions also. For that we have a data set here that is known as heterosis. Okay, so if you will view this data, what kind of data set we have, we're having a different kind of lines and testers. So this is the place from which they have been taken, the application, the treatment, the factor, and we have male and female combinations here, which are very much required. We have different kind of variables. For that, if we will use this function, and we have to replace the name of the data set, simply click on the enter. So we have the line into test analysis of variance, then ANOVA with parents, then ANOVA for line to tester, ANOVA for line to tester, including the parents. Then we have a GCA effect, SCA effects, and we have some other genetic components and a proportional contribution to the line to testers also. Then if you want to perform, say for example, path analysis also in this, we can perform the path analysis also in this. For that again, we using these kind of codes, or we can directly use the data. For example, we're using these two data set. Data is on build and soil, so we can perform this path analysis also. We'll be having the residual effect and the diagonals. We have direct effect, and above and below we used to have indirect effect. Similarly, we can also perform the ME analysis, which is a very recommended analysis for multi-environmental trials. We want to see the stability of some genotypes. For that, we'll be using a data PLRV, which is by default present. Then we can make some biplot, triplots also. Say for example, this, will click on this, and we have these ME with respect to principal component analysis. And again, if I will go to this, we can perform some other stability analysis also, like we have here. Let me show you what kind of stability analysis we can do here. Say for example, stability parameters we can estimate. For that we will use this type of a data set, and simply you have to replace the name of this data set and click on this enter stability. You'll be having these kind of analysis where you can see whether a genotype is stable or not with respect. So to perform the basic analysis of variance and experimental design which are used, we can also use in another library that is known as AgroR, a very important library as far as these factorial experiments are concerned, as far as these in these block designs are concerned. For that, we will use some functions here, like interaction plot, transformations, and all these kinds of plots. Plot dot interaction is very much used whenever we are doing and performing the analysis of factorial experiments, where you can beautifully represent the interaction plots which exists between different factors. So we have this kind of plots for factorial experiments also we can put. So these type of libraries, which was a quick review about which of the libraries are available in our studio, so that you can put your data in utilizing these library, and you can save your time instead of using some other kind of package. These packages are highly recommended, especially for experimental designs. Hope you like the video. Thank you very much.